Should we start? What do you think, Natalia?
Yeah, I think we can start.
We are also went live on Facebook and everywhere. Hi, everybody. First of all, we are very excited to have you. And if you don't mind, just turn on the video so that we can see everybody, students and who is just joining us for a lecture. And thank you for choosing our workshop. And I hope it will be like a really exciting and nice week kind of for all of us. I'll start the lecture, but in the end we will have you can ask questions and we'll be happy just to kind of talk a little bit more about it. Can you see my screen?
Yes.
Nice. So we're very happy to be a part of Digital Futures workshops this year and kind of share our approach and our research that we're doing with Natalia. A little bit about myself, like I'm an architect and an artist, and I work with biodesign, with computational strategies, and robotic fabrication. So, wait, I'll just hide. Oh. Okay. Like 5 years ago, I started like an architectural association with Michael Weinstock, and I did MTech. After this, I also joined Strelka Institute in Moscow for more AI-based and speculative design. And after this, I worked with the K Innovation Lab, and it's kind of really formed over the last years, like a research and directions that I'm doing. So I would say that I do believe that to start kind of from like a core ideas, or that's like the drivers of all projects, I do believe that people are always learning from nature, and it depends. Sometimes we just replicate it as processes, like in terms of what are we growing, kind of how do we grow, how do we cultivate pearls or anything. Like if you look a little bit closer, kind of on granularity of things, that's everything. It comes from like nature-inspired design. But it could be very different in terms, sometimes you can just try to copy a process, and sometimes you can try to like really collaborate and use this kind of intelligently. So if you will look on a different layers on Earth, kind of, we just see the similarity and connection, kind of connectivity of all elements. And we can see that's like, I don't know, spider structure, or like if you look into Mazelum or like snowflakes, it's all kind of in one case repeat each other, at the same time completely different. That's like complete, like I don't know, Mazelum overtakes the Earth and kind of stay there for a long time, and ice flake like will exist just for like a moment or like longer, it depends on the temperature, but there is similarity in structure. So kind of you try to find those patterns, those kind of segments that could be useful for you as a designer. And I do believe that it's time for a shift between like Anthropocene and biocentrism, or kind of finding out a different place in like a kind of whole bioweb of a lot of participants. And it really depends which kind of players, how do we play, like that's kind of what's happening with our like planet. And maybe it's enough like to kind of abuse and overtake the resources and start rethinking like how could we use it completely differently. And I would say that it's time to think how to create a kind of flexible, intelligent spaces, and finding a balance between who we are as a humans, like because we're biological kind of creatures, and how do we live like in this kind of hard, concrete like world. Because maybe it's time to start building differently in terms of like layers and functionality of structures and flexibility, because after one generation, like buildings, they don't perform exactly the same. That's if it's like a concrete structure, it's really hard to recycle. But how to make these systems like intelligent, able to adapt, to move with us and be like more like a digital kind of things that we update our phones or update our software. Can we update like the buildings the same way or design them initially like more flexible for like a future generations? So starting with the LIFORM, Living Megastructures, it's quite a big project currently that has a lot of different branches and different directions. And one of the main core, of course, like collaborating and working with Mycelium and looking into interspecies collaboration between human, robots, and like Mazelum. But at the same time, this design approach could be transferred in a lot of other biological systems. So it's mainly to trace kind of the whole pattern, like how can we like kind of approach the organism. And one of the directions here is physical experimentation, and it's growing Mycelium as a different media, like growing it's in the air. So it's slightly different. That's like a usual approach when people grow it in molds. And another one, like actually what we will be looking during the workshop, how to build after, like you see the results in the physical experiments, how do you build a computational logic with it. And another one is also looking into like how does it function and how to be like a devices, like performance-based devices that has Mycelium at its core. And we talk a little bit more about more design applications that we developed with Space10. But I think the main thing is that it's more like a bigger picture, that there are small fragments that we kind of try to grasp and just think about how it can change or like what could we implement and which kind of aspects we can understand to use it as a design strategy. So everything started in 2017 when I graduated from MTech, and I really wanted to do a robotic fabrication workshop to work with robots because my dissertation was there, like a multi-robot system. But then I understood that like you can do digital fabrication, looks cool, but if you don't change the material and initial media, kind of you will be still kind of replicating. Maybe you will kind of get the new geometry, but you won't change the core of everything. And my goal after this became like how to look in the material and how we kind of design the whole fabrication method. So basically with a startup that I was working on, we were looking into membrane and how can we create like kind of surfaces, more like thinner like membranes, but they didn't have like a real structural performance. And lately I understood that I'm extremely curious into more like design application and more like creative aspect, also like my personal like kind of nature. And now we will go a little bit more in the branch of like design application, and then we'll go back to like more kind of futuristic vision and computational strategy. So Biofold, it's a circular design exploration with like Fracta back and Space10, EKS Innovation Lab, and Thomas Clavijo. So whereas the core was circularity, like I think like an all sustainable project you decide your set of questions, what you're not you don't want to negotiate, like it can be any more the same. And here was first question goes back to my question after my dissertation, that every project that you do, it's like physical project, but even like actually digital, has its materiality. In terms of physical project, you always kind of build a whole story where material is coming from, how do we produce and recycle it. So kind of you need to ask these questions first before picking up like a kind of fancy cool material, because it's extremely hard to recycle. Construction, furniture, it's kind of the worst industries. But then even if you build digital world, it will be always kind of inspired by the physical world. And I think it goes back to my first question about finding a balance between digital and physical. You kind of look for this combination of things, but also how to use like digital tools, for example, to produce from physical materials. So here, after we defined that material is a core, we started to look into what's around us. And one of the main thing, I mean, we joined Space10 with an offer like to work with Mycelium. And then Mazelum really likes like fibers and kind of agricultural waste. But what the matter with agricultural waste? Because no matter where you're based, it will be like there are like local agricultural waste, depends of your if your country grows wheat or rice or corn, it will be different fibers. But because our city is like growing, it's simple that we need more and more food, so we will be having more and more agricultural waste. But currently just around 10% is used. So we have this amazing resource that we all need kind of to start thinking how to use. And also like it's a huge problem for farmers because they even pay like who could take those fibers, because it requires a lot of water, a lot of nutrition, a lot of problems to recycle those fibers, and the amount is that it's megatons of it. And also in countries like India is a big problem that they just burn it, and some other countries, but it creates a smoke and a lot of problems for people like to breathe. So for not just for this project, but if you have this amazing resource and there are so many problems with it, like, so it's a great idea to start thinking how we could digitalize it. Nothing new, but another thing that we put on the table and that we understood that we're like really worried about it, that's the amount of plastics, but not like how to kind of destroy it, like or like burn it, or like how actually to use it as another resource, another medium that we can do something with. And of course, like our questions about how do you recycle furniture and crazy amount of furniture and construction, like how to recycle a building. But if you like, for example, a student somewhere in a new country, you just get like a case table, you use it for a season and then you throw it away. So what, like, does it look normal like for you? So we kind of found a question that we were fascinated with, and it's how, like, what if we could produce furniture locally, having positive impact on people and the planet, and how we really can change it for next generation, how we not overproduce, how will we think the whole furniture fabrication strategy with our skills, knowledge, and ideas. So we developed a technology that's initially what I said, like started with Mycelium as a binder, but as a lot of, as the most like fabrication Mycelium kind of method, because when I was starting like 4 years ago, it wasn't so popular. Now it's like blooming. Everybody kind of wants to experiment with Mycelium. It's great. But the thing is like keep growing and mold, it creates a lot of problems. That's you just need to create a mold every time you produce it. And also you need a huge storage. So kind of really limits the fabrication. So we started to look how can we change it, how we can create a sheet or maybe like do something with that. So how do we play three-dimensionally? And we developed like a method that you compress it with vacuum and it keeps the substrate together. And it's very challenging with biocomposites. You need to compress it. So, and it's also anybody at home can can do it, just compress it with your vacuum bag. And then we were like able to create kind of fun geometries. I think it's like 4 a.m. in Copenhagen that we were like playing with. And we were like extremely fascinated. Cool. Doesn't look like anything like useful, but like the shape is cool. So whatever, how we systemize it kind of and take it further. So, but then another question came into the game. So using a lot of plastic bags, like, do we actually sustainable, guys? That's how we kind of can rethink it, like, because we saw like what's happening in our studio with all these plastic bags. And they are like a kind of a very new thing came into the game, like a cultural approach, because I do believe that there is no technology without a context. So to make it relevant for people, you need to make it attractive. You need to make it kind of fit in existing infrastructure. So we decided that IKEA bag, everybody has it at home, and usually like pretty much it's everywhere, but how can you use it kind of as a media? But also there is like a really crazy thing, like when you have free time, just Google like Fracta fetish thing, because people did everything from Fracta, like from underwear to sneakers, like everything. I don't know, like what's kind of motivate that, what was the motivation? But initially Fracta was one of the first things that was reusable bag that's released like more than 30 years ago as an idea. Oh, sorry, I just closed the window. It's quite loud outside. So yeah, and we decided like, okay, it's maybe also will be a culturally like attractive for people to use. So we got like really excited by like, how do you like kind of hack IKEA's bag to create, to use it as a media, as a like device to kind of form and shape and grow furniture. And we started like, and also like kind of with experimentation, we really saw like a people's feedback that it's like really looked exciting and interesting for everybody. So we developed a set of instructions that you can use, just kind of use tools that you have at home to create from 2 fractals kind of your device that you'll be hacking. Not like hacking Fracta, but then kind of creating a certain like geometries. Of course, like on the diagram, it looks easier than like in real life when you actually need to produce it, but it's still like a doable thing. But then let's go to the core. Like the core is material research. Without sustainable material, there is no sustainable like product. And we had a lot of problems with this project with the binders because to solidify inside without air, they're like, it's quite challenging. It's just like a usually chemical, like artificial binders can do this. So we were like went like wild with experimenting with a lot of different resources, fibers, waste, and this time we worked at in Madrid. And so going back to Mycelium, Mazelum didn't like being without so much air. So it was really hard to grow inside. So it was just creating a beautiful surfaces that we just wanted to keep alive because it's fresh, nice, like attractive. But still when you dry it, it doesn't have structural performance, and it's also inside, it's quite weak compared to like mold or anything. So we experimented a lot with plaster, but plaster is not sustainable. Like it's sustainable if it's pure, but there are still quite a lot of problems with fibers. We went in a lot of experimentation with different bioplastics, but after this all, we were very lucky to team up with a UK-based startup who was developing, and they keep developing like 100% sustainable like binder, like a resin. It's extremely amazing because you can eat it, but they helped to develop formulas that were able to solidify inside the plastic bag. We still like need to have more experiments in terms of like structural performance, but this was like kind of aha moment for us, that it was like doable, and also that you can see the fibers, you can see where it's coming from, which fibers you like worked with. There are like it's not just a like pure kind of surface of the binder. So also like going back to aesthetics, like to kind of find this unique, attractive thing, because it can be amazing technology, it can be amazing, even sustainable strategy, but still people kind of need to get attractive for like designs that you do. And but going back to like design strategy and like the core of it, that's and I think it goes for every project, like again, that's you develop like the whole process, not just like, okay, where's like my customers and like what they will kind of get and buy it. How do you grow it? How do you fabricate? How you use it? How do you recycle? You design all steps kind of. And I do think that's it can be different anymore, like that you just release like products without this. And also it's nice to kind of make things available like to people, that there is no overproduction. People produce when they want, when they kind of desire things. And also if they don't have any particular skills, because parametric design is fun and it's the future, but like maybe it's not available for everybody if you don't know like software. So we aim to develop like, to develop like a platform that's like people can just go and kind of get the design, get instructions, and repeat it at home. It wasn't released, but as a like kind of strategy. It looks very simple, but like it took us quite a lot of iteration how to simplify the logic project, process and ideas that people can just feel that anybody can do this, that it doesn't look like complex, like coding or something. And then we, like for our release, we designed like some pieces that kind of can be aggregated, because Fracta has a limitation in size. It can be aggregated and placed anywhere. So I would say that thinking circularly means taking the complexity of the industry, but at the same time, using the advanced technology we have, advanced knowledge, but at the same time, maybe to rethink like who we were as a people like 100 years ago, when we were closer to like nature, earth, like how do we feel in the city? So kind of shift some things and think like where with our capitalistic kind of approach, where things went slightly wrong. And going back to Lform is like still like the main direction is much more artistic than design application. It's more like as I said before, like a fragment of like a megastructures and more like kind of future thing. So was like an exhibition in Moscow, where was like a Bioincubator was installed, and during the show, like it was growing, and people were able to see the progress, how the structure, like how it's kind of how Mazelum overtakes it, and which kind of things are happening. And the feedback was like kind of really great, because people really see that in one case, it can happen without a human. In the other case, it's still Mycelium kind of into the game. So Mycelium adapts extremely like quickly to the environment, and usually it grows onto the ground, but now it's in the air, creates a different morphology, different functions, and that's what's like we will talk further. But how do we measure this? Like, because if we want to like kind of take it further and kind of break it like into like segments, logics, but like a Mazelum is like huge, like you don't know like how to extract this data. And that's kind of what was like probably last year about like looking into computational and also the result of the physical experiments. Because when Mycelium is growing, it's kind of has a calcium, it's pulsate, like kind of there is like pulse inside, and it moves very like quickly, but with changing one or like more conditions, it will be keep creating a different results. But also like when you think that it's just no matter how big it, it's all connected, and there are in every cell, there is like water, like it's transferred to all systems, and there are, for example, proteins that let water in and out, and that's what it creates this ability to collaborate with plants in exchange for like food. Mycelium kind of gets delivers water, and it gets kind of back some food, or like creates a connection between trees. So it's also like looking how Mycelium not just kind of exists, but also how it's connected to the whole system. And that's one of the experiments, the image of like our workshop, and it's one of the experiments that the geometries that we got quite fascinated, because what's amazing here, first of all, Mycelium built the connection between food. It's completely Mycelium generated. There are no direction for it. It just because it's in the air, it's got a new opportunity. It started building not like more like 2D structure, but like it started to create like a 3-dimensional. Second, because it's has a very different strategy of overtaking everything. If you see like pictures on the right, there are like two. It's more like kind of when spores overtake and attacks and moves as fast as possible to overtake as much as possible. Then the two images like on the left and in the middle, on the bottom, these fluffy things, it kind of collects and gets the water in the system because it's needed. So it's kind of created a new thing that it doesn't use under the ground to get this water. And then it's kind of thickens and rebuilds the structure to actually start getting like this morphology, this resilient kind of, and quite a strong system, because I have dried samples and you can touch them. They kind of keeps this geometry, and where it's like transfers water and nutrition through the whole bus. But then another cool stuff happened that, I don't know, does it look for you like mushrooms? But it is like, it's a eatable mushroom that just because it was in the air, it didn't have enough microelements, and probably it's not the same like soil or like if you grow it on wood chips. So it started to create these deformed bodies in the end of like when it's kind of overtake the whole experiment, it started to create on the top, but doesn't look like a normal mushroom. It looks like more like Lachen. And Lachen, if you know, it's the mix of fungi and algae. So it's a kind of course, it's not like exactly a mutation, because we have a lot of conversation in terms of like, if you keep transferring one mushroom from incubator to incubator, will be it's adapt faster. But in reality, it's created a new structure that's Mycelium, like usually like it creates a fruiting body, it creates a mushroom, but here it's became more like algae fungi from the forest, but still in the incubator. And it's extremely like also fascinating that it seems that it's more like an optimal like geometry is the best path to transfer everything and microelements for it. And another cool thing, it's functionality. How does it function and how does it get kind of water? And for example, in the incubator, it will get all drops of water and it builds kind of connections, like a small like structures to get everything everywhere water is like gets collected or like on the bottom. So it will be kind of really getting to get those drops and build the most efficient path for it and structure, and kind of also the maximize the surface and bring it back to the system because it's still one body. And I think like an amazing example also like for computational logic and inspiration. This is a mushroom root together with plant root. So it's mycorrhiza, and it's more like the interaction, how strongly they interweaved in each other, because it's hard for a plant and the mushroom to find exactly the same type of like species that they collaborate. Not all of them are friends. But these plants find kind of the right mushroom, and then they start to exchange nutritions, water, and it's crazy how close they are, like as kind of close bodies. And I think like to start breaking it into computational logic, we can look at this protein because it's like a high resolution image that in 2017, researchers got like a Nobel Prize for it, because on the right we see like the granularity that they achieved to documenting proteins. But currently, like in MIT, they simulate and they use machine learning to generate like protein structures based on the knowledge that they got from these images. So going back to like Mazelum, we're like thinking it's extremely challenging. Like, how do you not just transfer kind of and try to make mushroom-looking structures? How do you simulate and look into design process, and how do you extract the core things? And as I said before, so just to kind of combine it, what's good for us for the workshop, but in general as a design strategy to take for any kind of system design that we learned from Mazelum. And one thing is the intelligence of the system, where every cell is functional. Like no matter which cell you drop a chemical, like it will be everywhere. It transfers, it functions and breathes, it transfers like water, and there are like channels. So all, there is no like a separate thing like a concrete and then another layer, another, it's everything is functional. System has extremely different strategies when it's overtakes the environment, depends if it's in the air, which kind of humidity around, like how does it get water, how does it maximize the surface, and then how it rebuilds the structure to make it functional. And yes, it sequence kind of all important networks and process. And yeah, that Mycelium adapts. It will adapt to anything. Like if you just provide it with the right nutrition, water, and microelements. But it also can stop if it's kind of used all resources. But if you don't kill it, like it can overtake it again. So it's also reactivating the system. And that's like a bunch of kind of elements that you can just take as an inspiration and as different strategies, as different logics and the combination kind of it. That's with extracting it and studying this, that's when you kind of start to understand how, because the complexity is just kind of crazy. It's like how fast it changes, how crazy it adapts, because every test is slightly different as soon as you changed anything in the combination. And it's kind of going further, more design approach. This is from our another project like intelligent, from intelligent morphology, like IM project. That was a functional like kind of system and high level meta, the name of the project. That's more like how do you assemble the elements, that's a function that keep transferring elements, and how they combine kind of functions in the structure, like in the exoskeleton, but at the same time it's transfer water, it creates like possibility for plants to grow, or like wherever is happening. So kind of how can you build systems, but also that you can use as an update for existing buildings, or like a separate structure and design strategy. So I think this is it. I hope like it's quite clear what's like advantages of like Mycelium as a media, but also as a matter, and kind of which kind of what's like in terms of design approach you can take for using other biological like creatures, organisms, and at the same time how you can start transferring it more like computational logics. Yeah.
Okay, great. Thanks, Katya. It was interesting for me as well. And I think we have 2 questions so far, and I think if you have more, you are encouraged to ask, and we'll be happy to answer. And I think Katya will be answering right now because she's more keen in that topic. So the first question is, can you please explain what incubation is like for the mycelium, especially for the large scale installations?
Mm-hmm. I mean, in one case, it's simple and at the same time complex because it's quite easy to make mistakes, and I had experience with it. But basically, in one case, it's glass or plexiglass cubes, kind of the main thing to protect mycelium, because it grows under the ground usually, and it's not a perfect environment, but it's extremely hard to recreate it in the lab in terms of like to keep organism alive, because when you create the environment, what mycelium likes, it likes humidity, it likes warm, and like there are some other micro elements that just want to jump in, like mold and stuff, and they're all stronger than mycelium. So kind of you really need to protect the good guys. So you need to keep it clean, like sterilize everything before the installation, and then I have in the exhibition spaces inside, there is also like a closed type UV lamps that don't kind of affect mycelium during the process, but protects and keeps the air kind of clean. But it's for like a bigger installations. But for the smaller one, it's just like kind of setting up everything clean. Then also like, how do you keep providing it like water for it? Because it's kind of will overtake the fibers, but it needs to have enough air. So it's still like some air needs to get in, but at the same time like water. So on the big ones that you saw on the images, it was like air humidifiers inside, like under it. And then it was like lamps on the top of it. And yeah, and next week, like I'm opening a new piece. It will be like a water, not like air humidifiers, but like it will be like a water just on the bottom incubator, because my next study will be about in particular connections and how it gets the water and pump it up. So, but basically incubator itself, it's more like a shell. It's more like an envelope to protect it.
Yeah, thank you. And also I can add, because I was experimenting with Katya once with the mycelium growing cells for the inhalable matter project. And I think after 3 weeks, maybe growing, there was everything. Like not only mycelium, but there was like a forest, a bunch of microorganisms, but it was still very beautiful. But the structural performance, of course, reduced because of that.
Yeah, but it's like the elements that I send you like as a sample, and I was just like, should I send it, like, because like when exhibition space they requested it, but it's the piece that was like slightly imperfect. There was like a touch of black mold, and Natalia just told me like, it's kind of cool. It looks like a real things, like with a lot of complexity like inside, because I try to do everything perfect, but like that's actually what I'm learning a lot from the organism, like the beauty of imperfection, the beauty of natural surface. And also I think I didn't mention this like in the lectures, that when we talk about artificial and natural, computational and kind of natural, it's we're in a very interesting place as humans, because we got used for these like a perfect petroleum based surfaces, but we like as a biological creatures come from like, I don't know, like touch, like how do we feel in the forest, how do we feel like when we kind of touch the trunk, but then we come to our perfect white spaces, and it really changes a lot in me, like kind of accepting imperfection and beauty of nature.
Yeah. Okay, we have one more question. Do you guys have ideas on mycelium projects to have structural performances that could be implemented to macro scale, such as an architectural project?
I just wonder, what do you mean by micro scale? Because
macro, big scale.
Okay, because I said micro, I was like imagine like
no. It's my pronunciation, I guess.
No, your pronunciation is great. I think like it's in one case, like LIFORM megastructures, it's already a dream about these cities that grow and that kind of overtaking. But if you talk about like actual physical project that's like, I don't know, you can use tomorrow, possibly, maybe. It's the same like with like Space 10 that we did like more like a study on the Mycelium that you can, because initially we wanted like people just to grow their furniture at home, to like deliver incubators for them. But I think like some people are extremely interested in like building like, I don't know, bricks or something that you can use right now. Like, and I think the more purpose of this research is kind of look a little bit further than this, but possibly maybe.
But what about the Ecovative project? Ecovative, they are building the big structures, right?
Yeah, I mean, actually, I just saw like like a few weeks ago, like a new project by David Benjamin, that and he was like really from like discrete elements, kind of letting them interlock and to grow in each other and kind of creating a big structure. No, but I thought the question was like about us particularly. But I mean, in general, I do believe that it's possible, and also I'm now really fascinated, like how can you transfer information and everything with Mazelum networks? How can you kind of really extend this performance? But it depends. Like you can build bricks. You can build depends how what's kind of the goal.
Well, I think the main problem is that Mycelium doesn't grow really well with the light. So it needs the dark areas to grow.
Yeah,
it still grows, but much slower.
It's true. Its growth is much slower. Within like I had an experiment with light, and it's really slowed down everything. But now I can't talk much, but we're developing like a solution that's hopefully will kind of let this thing work. But I mean, with Mycelium, like with every biological system, it's a lot of problems. Like if you take bees, like amazing structures that you can create with bees, algae, like also great. But all of those systems, everybody got stuck. Like with how do you
scale up?
And scaling up is the hardest thing. For example, with startup, we were looking more about how do you create boards that you actually can replace plywood and you can start CNC and fabricate things. This looks much more like closer to reality than actually let it grow. At the same time, over like last years, I'm personally shocked that's like amount of progress. That's like there were a lot of things that when the first idea was like circulated, let's grow it in the gallery in real time in the incubator. It just looked impossible because we were like experimented with Natalia with plastic bags, like everybody, and grew in it, and it was great. But just the idea that's like it's the body like kind of can be naked without any plastic protection was just terrifying. Like to kind of try it out. And and it's so good.
But anyways, I remember the exhibition that we did in the gallery when not the same project that you were showing, but we had an experiment before that. It was not that beautiful to show it in the lecture. But anyways, the most fascinating for the visitors was that it is growing right now, because it was the cube with the mycelium shape inside, and it was covered to protect from the light in the gallery. And so the users, they can just look into the window, and it was just exciting for them to it's like you are looking into someone's door hole, because you see some process that is going on and it's moving. So I think this kind of a game maybe that we implement into architecture and design is really exciting, because this is what nature does always. It just flows around you with its own processes and its own laws, and we are just we just need to adapt to it or watch it. So I think that's amazing when we use this knowledge in design and architecture as well.
But I think also like it was amazing the connection, the experiment what we did with from IM for like where what I showed in the in the end our kind of grid structure. So there were units interconnected with Mycelium. It was like more mycorrhiza, like more like a combination like for like roots, and how the plants like were like kind of they were growing better and what's kind of building like a system, because it's another our big research with Natalia about trees and plants and how they communicate in the forest and how do we transfer it back to the city, and not like kind of just drop separate trees that they have like a shorter life and live completely different compared to the forest. So kind of how do we rebuild and interconnect them and create them, even if we build it in I don't know, between skyscrapers and the dense parts, but have like kind of keep the same like with Mazelum. In incubator, you need to keep some conditions and you can break some. So, and with the plants, we try to keep some conditions to connect plants that you like, not like in all green walls, you need to kind of reuse those plants. How do we create an actual forest, curated forest, but happy forest kind of inside the city?
I can also suggest that maybe you can turn on your mic and ask the question if you like, because we are now more in a discussion than in a monologue. So yeah, because we have one more question, I will read, or maybe Jacqueline, you want to say yourself? Okay, maybe I need to
unmute them.
Yeah. Just a second. Okay, I think you can turn on.
Yeah, thank you. Do you hear me well?
Yeah.
Yeah.
Okay. Just, I was just wondering if this need of darkness is the reason that your project start like in the interior design, like in furniture, rather than the exterior, or it was more like a personal approach? Because well, I'm studying architecture, so I was thinking about using these structures like a facade, for example. So
you're using for what?
For facades, for example, like the front view of a building.
Mm-hmm. I mean, in reality, like because it's a like research, so I'm like fascinated that we keep breaking the rules. So I assume you can find a way to grow it like in the dark, and I don't know, depends on the glass or one. Unfortunately, I can't talk about the new system yet, but that we were developing. But in one case, it's the same like with micro elements. That it's like small wood chips. It will be stronger on wood chips than on the fibers that's hanging in the air. So you always kind of negotiate. Like I'm interested in the structure and how it grows in the air, but when I want a better structural performance, I will go back to like I don't know, wood chips, or I need to find how I kind of implement more micro elements in the fibers to make it stronger. The same with the darkness. Maybe you can stimulate it much more with a different kind of stuff in the fibers that it will eat it so good that it will kind of forget that it's light on. Currently, I had like not the best experiment with this, but I do believe that this kind of could be overtaken. And also probably there is like a percentage of time that you, for example, in the gallery, like when somebody comes in, they turn the light, they see it, they turn it off when they leave kind of like the gallery space. But it's most of the time it's closed. So it's much harder, for example, now for a new piece like in the museum, like when it's constantly, like because the first big piece, it was like in the private gallery, and now in the museum where it's a lot of people constantly coming. But I do think it depends if you want to have it alive or you want to have it dead, like already, like you can like grow it in the lab and it's dead on your like facade structure building. It doesn't matter for it after it's killed. But if you want its life, if you want it to function at the, I don't know, network at connections, then you think about like how do you protect the body, or like how you maybe even genetically modify it. Depends. There is like, I think, unlimited like speculation about it, like what can be done. Because I don't know, mushrooms that you buy in the store, like they don't get brown most of the time. Why? Because they modified. So maybe like nobody were curious about how to modify it, like that it will grow kind of in the light.
And also, I think in the light, they start to produce the fruit bodies, right?
Yeah, but I think like what's sure that it's more like the speed. It's like the same like temperature. It's like to get to a fruiting body, like kind of it needs some time to kind of develop before you get this. It won't get just a fruiting body like immediately.
Mm-hmm.
It was actually very interesting was the last test that it's needed to overcome everything and it's produced these deformed fruiting bodies, but it was never exposed for light. It was always in the dark, but because it's kind of got like, I got all food, I got to produce something. So it's got like these fruiting bodies, even weird ones.
I think anyways, the material research is not limited to the mycelium, because like in the Space 10 project, you just switched to biocomposites, which was also, I think, suitable.
Yeah, but I think you get the inspiration, like I think from Mycelium. I think I'm so many times I give up on this project that I'm not coming back, but it's still, I always have like something that I'm so curious about that I come back, just like, I'll just research this and I'm done. I'm tired with these experiments with mushrooms. I'm tired that all my friends send me all videos or like all articles that's mushrooms on Mars or anything. I mean, I love it, but it's like, you can't imagine like how much stuff like this I get. But then I'm like so curious, and then I get so fascinated, like because I shared so many pictures when we looked with Natalia about these deformed bodies, and I was like, this is the beauty, like this is a geometry, kind of how can I create this way, like. So I think it's more like a learning process, and then think about how you use this strategy somewhere else, like with Mazelum or without, or even like if you go. Well, during the workshop, we'll be doing like playing more with like a big scale, but still inspired by the knowledge that we got like from small.
Yeah, exactly. Like as for me, I'm the urban designer, and when I saw those pictures, I instantly said that, oh wow, this is the city scale model. I said, no, this is the materiality. This is the Macroscale. Yeah, so because it is really amazing that the structure is applicable to all scales, and this is the same material which performs differently. So I guess I hope that we can develop this thoughts during the workshop, and I hope that we can produce some design strategies and some really cool shapes based on those knowledges, because even for us, this is the experiment, the ongoing experiment, and we learn from those structures, and we hope to learn from you as well. So we really think that it like every person has its own ideas and its own personality and design personality as well. So it would be really interesting for us to see what you can do with those inspirations, let's say, and how we can came up with the design that can combine all those interesting ideas that we all can produce. So this is the most interesting part of these workshops, workshop for me.
And I think it's extremely hard for like grasp kind of in biodesign and biomimetics to grasp kind of idea, not to copy the organism or not to copy like idea, because I think it's the worst design when you see like, I don't know, something that looks like a leaf, like actually having a functions of it. So it's but it's still like, I remember when I was first introduced like in a to those ideas, I was just like, so what I'm doing with lobster, how do I kind of break this system? And it took me the kind of some time to start not like a copying kind of a form, but actually looking how fibers are arranged, how can you kind of design the system like from the same material, or like how do you scale this up with some taking some principles. So it's not like that you need to take everything, you just take like certain aspects. And that's why I showed like a project with space-time, because it's just a default branch, but it all comes from like some knowledge about mycelium that goes kind of different directions.
Okay, maybe you have any more questions? Let's see. What?
I'm curious just about one.
Hi. Oh, sorry. Please go ahead.
Sorry, Zaba. Is any difference like in the sensation between having the live or the dead structure? Like for example, my question is like if it works like in other plants, like it gets colder the weather when you have like living plants around, or it doesn't change?
When it's alive, or like,
yeah, you told me that it will be like a decision to have the structure leave in the structure or make it dead. So it's a difference like in sensorality to do this.
Yeah, it will give you wait, sorry. I don't know, this one is, I'll just show you, it's like for like, it disappears, huh? Oh my god. That one show you.
We saw something at the beginning.
Really? No, but it's crazy. It doesn't kind of find.
Oh yeah. Just now, instead of your face.
Oh
yeah, that's.
Oh yeah, I got the point. How it's so. This is already dead for like, and most of the pictures that you saw, like it's from this one. So it's basically dead for last few months, and it just like there. So you can put it. I mean, outside will be a bit hard. You still need some coating for it. But what amazing things, it's like as more mycelium you have, as more it will be like fireproof, because mycelium is like, it's not burning, and even it eats wood chips or like fibers that burn pretty good. Mycelium is quite good with this. The same, most of the biomaterials have the problem with humidity, and mycelium is still quite good, like hygroscopic kind of properties. So it has a lot of advantages, like because the same like why people try to do laser from it, and laser surface, there are like quite amazing research, and brands like, I don't know, like Hermes already like kind of announced that they will be producing like bags from it. So what's amazing about mycelium is that you can engineer it in anything. Depends on the technology you use, or like, I mean, shoes industry is quite interested in. So it's all much simpler when it's dead. It's a material. It's like fibers, like dry fibers with like binder. When it's alive, it goes back to the incubator for like, how do you protect it? How do you keep it? Like, do you want try to achieve like that, like when it grows under the ground? So it's you don't like, nature doesn't do anything, like it just let it kind of be. But for humans, for like directed, for gated, it's like a very different game. So if you want to keep it alive, I do believe it's doable, and I know people who are doing research into like more electricity kind of transferring through the membrane, and more like because it's because of the nature of long cells, it can transfer like electric like signal kind of inside of it. But you will only think how to keep it alive, how kind of also for people, like how do you feel comfortable if it reactivates? Because if you just dry it, you can spray it with water and it will get active again. But if you dry it in a, like, depends on the type, like depends on the species, but more like than 60 degrees, it's dead. So it's dead forever. So there are like quite a lot of decisions to take. Did I answer your question, or I just confused you even more?
Oh yeah, I think it does. Thank you.
I just really like to confuse people.
No.
It's like, I remember somebody at school told me, like, convince them or confuse them.
Okay, maybe Saba, you want to?
Oh yeah, I think my question has been answered. I wanted to ask, like, how do you make it dead? And you did answer that. And also, once it's dead, can it be like placed outside as a sculpture or something or of the sort?
Mm-hmm. I would say, like, in a sunny day, yes, but to leave it in the air, like, you will still need to coat it with something. But I was always, for example, with the same with Biofold project, even with biocomposite, like, it's better to keep it inside. Because Mazelum materials are more like raw wood. If you throw it away, it's kind of will decompose. If you put it inside, it can stay for forever. But you can still use like shellac, like natural coating, or kind of other, like, so you will need to coat it with something if you actually really want to leave it outside forever. Do we have more questions? Quite. So we probably should finish this one.
Oh, sorry. I also have a quick question. Sorry to interrupt. It was a really good lecture.
You're welcome.
Thank you for that. And I have a questions on, do you guys have any future ideas or pathway that trying to combine myceliums with 3D printing methodologies?
I mean, 3D printing a bit less, but with robotics, yes.
Oh yeah. Okay.
That's
So
a new game. We're just developing like a slightly different approach when we try to go away from 3D printing. But there is like something that's like a kind of a new project with.
Yeah, Katya actually is participating in Tallinn Architecture Biennale with this project. So hopefully you can soon see.
Like some new stuff is like coming, but it will be very interesting. And also it goes back to the concept of like interspecies collaboration. How do you introduce more kind of agents inside? But I think in general, 3D printing is interesting with Mazelum in terms of, like, in one case you can create shells, and I think it goes back to like Jacqueline's, for example, questions that you can create like a shell, and then it's goes kind of and fill inside, and you can use these structures. In other case, much more, I would say, like kind of more fascinating. That's you 3D print with like micro elements. That's like will be food, kind of food and direct mycelium. But it also goes to the point: are you doing this in incubator to keep it more like safe, or like you kind of develop a method and to try to do it kind of outside, or you just 3D print, like, I don't know, wooden structures that you put like in the incubator. So there are also like a lot of space to research and a lot of kind of ideas. It just like I personally can go in all of them and just take my bits, but like I follow like I think it's Biolab in Australia. They like, I saw that they is 3D printing a lot with Mycelium currently.
thank you.
You're welcome. Anybody? Should we wait? No, for like a brave ones, or like
Curious.
I have a quick question.
mm-hmm.
You mentioned something about sourcing the mushroom from the supermarket. Those are genetically modified, that they don't brown. So is there a specific species that is most optimum for, to work with in structures and so on? Like, just curious about that.
Yeah, it's a very good question. But you basically choose your kind of players and your participants, because they eat different food. So for example, they experiment experiments with fibers won't work with like some other like species that work like just grow on wood. And they also, some of them like hardwood, some of them like softwood, some of them grow faster, some of them grow slower. So, but kind of build, some of them like have high amount of chitin, and chitin is in all kind of shells like calcium. So, and this one will be like stronger if you want to like have a brick. But for example, they won't grow if you will decide to grow them in the air, what I'm doing. So, for example, in the incubators, there's one that's like kind of big square one, or like all these kind of pictures that we showed. These all eatable normal mushrooms that you can kind of get anywhere. But it will be always kind of speed of growth, or like amount of calcium. You need to decide what the kind of your desire, what's the final outcome, because of course ratio are the strongest one for like structural performance. Eatable mushrooms, they have slower kind of performance than the woody kind of one, but they have like different options in terms of speed and in terms of like flexibility kind of. And also some of them more like kind of crybabies. They don't like kind of more things, and some of them more like I will grow anywhere, everywhere. And for example, I was really fascinated by the fact that if you put different species inside, you can scary the weaker one, that it will grow faster because it will be sweating and running like kind of away. So they're like so many things, like kind of like if you put few of them, which kind of food, so kind of you just have a kind of a full catalog of like decisions to make for your project.
Yes. Is there a catalog reference that you typically use? I'm just so curious. Is it like a spec sheet, or maybe do you work with biologists that sort of explain to you which ones are more suitable for which? Like I'm imagining like maybe there's stats, performance, speed, and growth, whatever.
I've never found like data like this. It's just experimentation, because in the beginning of the project, when we worked on the startup, I was working with an amazing scientist who is like material scientist, but also quantum physicist, like amazing friend of mine. And I was constantly calling him and saying that I need somebody professional. I'm an architect, I'm an artist, get me like a mycologist or somebody who's normal, who can do this, because I'm completely like lost. And he was like telling me that there is nobody, because this is a new area and nobody thinks like this. You can just go and do it yourself. And a few years after, I can say yes, it was just the one way out. Because with Natalia, we just had like a call with it's like a forest university, like as it's specialized on mushrooms, and there are mycologists there. But they were like, kind of, we are here to grow food. What are you guys talking about? What are you showing us? And we like just showing them images like, why my mushroom does this? Or like, why this created this deformed fruiting body? Can you explain me? And they like, because we're analyzing it, we're learning. And they were just extremely confused with us.
We are trying to grow food.
Yeah, we try to save the planet. We try to grow food. And we were joking that we're here to save the planet as well, just the way people be living. But basically just research. I mean, more and more people release their research, like wherever, like you can find samples that's like, I don't know, this person tried oyster mushroom, this tried reishi mushroom, and the amount of days it took, and the amount of like which kind of substrate somebody used. But if you, for example, want to do what I kind of do, that's to grow it in the air, like, okay, there is no research. You just I saw like a few projects that people kind of tried things out like this, but there is like a very small still knowledge, because depends on the questions I will ask. Like I will take one direction, but there is like a whole like set of kind of possibilities that you can research and gain information for like everybody. So it's really cool that people research and release. But really also, I think like, I don't know, patterns and like, that's more like people who try to do business are like, create some like, not for like artistic research, but like, of course, for startups and everything, there are still like active patents that's like, don't release, like don't let people do a lot of stuff. So it's also like kind of common desire, like to progress with the topic as a, like, I don't know, Mycelium material of the future, and like the one which will save us our planet.
I think that with this experiment that we worked together on, we tried to play scientists. It was funny. Because we did these experiments, we signed them up, and we were monitoring, and we were trying to do a schedule on the table and everything, but it was just a mess inside the
But we're architects, so we also tried to feed them with colorful stuff. They didn't eat anything colorful. We wanted like a pink, yellow, but like material, they didn't
work
kind of
at all.
react at all.
Yeah.
But still, like, it's just gaining the knowledge, like, and then see the results, because even small experiments sometimes give such a crazy result. And the one that's like even like currently these three-dimensional structures, but there were so many failures before. And I had like personally a problem with one gallery who was like, they were asking me if my mushrooms can grow faster. And I was like, can't do anything, guys. Like, you know, they're already there.
Like, I pray.
Yeah, I can just talk to them, like, and ask them. But yeah, but
what I wanted to say is that I think that this experiment just it told us really clearly that we are designers and architects, and we're like, if we do science, we do the architectural science, and we don't do microbiological math science. So we decided to collaborate with this Institute of Forest, and also I think what was important is that the next step was to develop those megastructures that Katya started to do, and I was really fascinated by one of her experiments because it really resembled her previous projects with different materials, like with glass or some really, really different material. But I was wondering how Katya made the mushroom grow her way. Because I think for me that was a really cool insight, because I realized that the person, the artist, the designer, it has some power. It has something inside that makes the material and the final thing do the final shape, or I don't know, in the artist's way. And I think this is the most important for a designer to find this, and to find something familiar in his works, and to understand what is common in all those works and why they are all made by me or by him or something like that. Because it doesn't matter, as I understood from Katya's experiments, like it doesn't matter what material she takes, it is her work.
Well, it's crazy. I've never thought about this. Like that my mushrooms look my way.
yeah, that was for me, like I was really amazed, like how she did it. It's the mushroom. But anyways.
But I think like it's in one case what Natalia said, that it's our weak part, that we are designers that try to do like biology, programming and whatever, like because a lot of computational students are really struggling that you don't have a programming background, or like bio kind of biology like background. But then in reality, it's your strong part because you was a fresh brain kind of come into the game, and you still have your goals. Because for example, I work like probably even more as an artist than an architect. But it's still everything what I do, like it's about the future planning. If it's speculation, if it's still about the space. I trained as an architect. It's done. Kind of, there is no way to go from this. And I think this is like, in one case, for me, it was a lot of like very hard like kind of aspects that you constantly try to find the support or help from somebody, but then I understand that their brains don't work this way. They like, for example, the other day I talked to like a scientist who work with the proteins, but they work with the proteins to create medical kind of different like vaccination or something, but I'm interested in a protein as a structure. So it's a beauty of modern architecture, kind of really accepting. In one case, it's a weak part, but at the same time, very, very strong. Because
yeah, exactly.
Nobody will think the same way. Nobody will think about how to grow a structure. Like in the University of Forest, they will be keep thinking about how to grow mushroom, like to feed people. But like then you come and say like, but I want a building. So.
Yep, agree totally.
I think there was like more questions.
Oh, okay. There is something. What are biological reasons or logic that mycelium or other bacteria grow into such complicated shapes? And how can they develop such intelligence even without the brain?
Beautiful. I wish I know the answer. Like it's more like a answer of like kind of the purpose of life. But I mean, Mycelium has, of course, like some like, and what we're trying to do during the workshop to extract those patterns. But Natalia knows that I'm sending her constantly like what I read in the book and what they can do. But one of the things, there's like a term homing, kind of when two different parts and different mycelium, for example, some of the species, some they can't connect, but some, I don't know, it's more like a 23,000 species that can kind of interconnect. So they grow towards each other and they lock to build this complex network. There are a lot of points when they start and how they kind of get together. At the same time with slime mold, it's a beauty of like amazing performance. Because if you take a Tokyo, like or any other city, like a kind of the cities who, like which has like a good infrastructure, and you place food on the main attractors and you release slime mold, it will rebuild the same network. So it will find the shortest way to take the food. So nobody knows like how this intelligent creature gets so intelligent without the brain. And I think a lot of researchers like ask this question. But for us, it's extracting the pattern there, like how it like works, like how it starts from different points. And also what I showed in the beginning, that's in one case it's different, like I don't know, spiders, mycelium, like it's different are like organisms, but then you see like similarity. So it probably works for a lot of them, so we can take it. Or like some of things like that's really hard to transfer, take it, or we are just not ready, but we can still keep looking at this direction.
Yeah, I think that to answer this question, you just start to go into some spiritualism always, because you can say that, oh, the spirit is everywhere. But yeah,
does Mazelum has a karma? Like...
No, but I think the same question is, how can my stomach absorb food, for example, without any brain, but it somehow it works. And I even don't control it. I don't know how it works, but it still works inside me, and I have no idea what it does. So I think there are a lot of things in the world that we still don't know. And I think that we are starting to open up, and the scientists world, not in the architectural, because this is not our topic, let's say. But anyways, we're starting to open up and to understand many things, how they work. And I always like to compare the biological processes and how we work with them, and also the material research and the shape in architecture and design with the programming. Because when you start, for example, Grasshopper, I think everyone here is familiar with Grasshopper. So when you start to work with Grasshopper, first, to create a really, really simple code, you need to use a lot of buttons. You need to create something that big, which is, and I think that like 99% of this is not useful. But then you work more and more, and you get more experience, and you refine the script, and then you can use only 2 or 3 buttons to do the same script. And I think it's the same when we, for example, are doing plastics. So why nature needs only 2 elements to create water, and we need, I don't know, 100 elements to create the plastic bag? That's because we are just newcomers. So I guess that we need to work more and to research more and to refine the script, and to work into the materiality of things, for example. And this the same with the other thing.
But
so
oh, sorry, keep going. I don't know.
Yeah, so I think that with the logic of the mycelium, so we just there are some experiments, and we just observe right now. We try to resemble the logic, and we try to make something that is trying to do the same. And we just learn from it for the moment. I think we don't create something new, but we still want to do that. We still want to experiment. And this is really important for a person to think about, for example, for me, as myself, like, what can I bring to the world as a person? What am I doing as an artist, as an architect? And what is inside me that is not the same as with the other people? So, and that's what makes it interesting for me.
But I think also, like, because all these organisms, when they kind of function and they go, they find an optimal way, or like kind of they have their reasons to be like this. And human brain is like complexity, like without a reason. And I think it's also learning how to simplify. Like every time I get stuck with a design, and because I want to create something, simplify it. Like simplify the logic. It will be complex anyway from like different kind of layers. So kind of keep the core, simplify it, and then you kind of can build like a much more intelligent system. But sometimes I think what you said about the plastics, like we create a complexity without like a reason.
Yeah, and with the shapes, like the shapes are complicated, but it's mostly the fractal. So it's the just copying the same pattern, and it makes a really complex at the end. But it's just because our brain cannot absorb all the information at the same moment. It's like when you start using Grasshopper and you understand that you can copy 5,000 shapes randomly with the one click. And before that, you would need to copy them. It will take ages for you. So, and your brain thinks, wow, this is so complex, this is so cool, but it's not. It's just copying the same thing. And I think what is amazing with the natural patterns is that when you see into the logic and into the process, it is not really complex. And it is really simple. And this is amazing. This is the most amazing, that it doesn't have any brain, but it's still, it does really simple things. And at the end, they perform like really complicated.
Or I think also one of the main things when you learn Grasshopper, you're like, okay, now I can make this crazy stuff. And then you're like, but why? Why would I? It's like next question. Now I know how, and I can like make these elements and I can like play with it all day long, but without like a purpose, without design, logic kind of and beauty of it, like there is no purpose in it, and this parametric kind of complexity. So I think also like what we'll be talking during the workshop, it's about like aesthetics and they're like clean aesthetics and like why is it important, like and why like I don't know, Mycelium structures are so fascinating, but they're really functional, that they channels to transfer water, or they have functions at the same time. This plastic but like kind of like unique morphology, like where every element is functional. So we're not doing grasshopper without just for fun to see something, or like print something.
Yeah, I think it's more about the design. Okay, so I think maybe you have something to ask? Something else?
I have a question. Sorry if it's noisy, but I have a small fun. And congratulations for this beautiful structure and the workshop. My name is Despina and I'm from Greece, by the way. And I wanted to ask, because I started studying a bit about fungi, and I wanted to ask if we can really actually simulate the growth of mycelium and predict how it will actually grow, even if we have a scaffold that is going to embrace. But nevertheless, fungi is something that has a life itself, and it actually doesn't have a brain, but it does have all these senses that we have. So my question is, if in your research you found something happened that you did not expect it to happen, and if it was something that grabbed your attention.
I think everything happens that like I personally didn't expect. In every experiment, I just look like, or like I opened the incubator every like 5 days, and I was like, that's something new, like. So it's very hard to predict it. You can like keep trying to extract like a computational logic from it and some aspects. And I don't know, in particular, I think slime molds or like they are really good with, people already kind of achieved some. But still, like if you change like conditions for Mazelum or slime mold, it's can start behaving like a crazy. I have a friend, like also an artist, and I remember like he was watching how slime mold in his incubator was like running around and like hiding, and he was just like, what the hell is going on, like, because it tries to like every day it's in a different position. So I would say to a certain degree yes, but like it also depends when you place Mycelium in the air, like there is something new start to happen. So this one is like, it's a keep studying kind of process. You can simulate it fully. And I think it's a good example. I'm not 100% like, I need probably to read more, but what I've heard about that's like Dolly, like clone that we all heard about, like, and always like a big breaking news. And then when I lived in UK, like it was like I think I was lived next to a place where it was like this research happened, and somebody told me, like, you know, it was like 5 of them after, and they all died from different diseases than Dolly. So they cloned Dolly 5 more times, and they died like from kind of different aspects. So you can't predict the life so good. We're still like far behind. But it's also what we're saying, the idea is not to copy it exactly. The idea is like to find how you direct and navigate, and also like a simulation kind of where it can help you to build much better and much more intelligent structures, and you will build yourself. Because of course, like if you even looked into the picture behind me, like it's really hard. Like if I just sit down and need to draw it, actually to draw it's a bit simpler than like you will go like a model kind of and do like a 3D model.
Simulate.
And then simulate, it's even like a new level. But still at some time when you reach like this level of the simulation, it starts looking kind of similar. but you still can play with conditions that you drop like a drop of water and it will grow differently right here.
I think I will talk more about that in my lecture, because what is important is not to control everything, I guess, and because it will be really boring to do experiments which you know that will grow and what will be in the end. So I think even when you, it's for us. I know that what we do in architecture together with Katya is a bit of about the future of space and future of what we can build and design. So that's why it's a bit specific. But what is interesting is to direct and to create an intention, let's say. So we cannot clearly predict. We can just make an intention what we want to have. And anyways, our life is a dynamic process, and everything around us is it's constantly changes, and it's flowing around us. And we cannot build something which is stable. Because even the stable buildings that they change every time, and you can see that. And why don't we put this into the design at the beginning? Why don't we really look into this when we create the design? And we can create, for example, the stages of our project or released project, how we can grow and how we can change with the dynamic forces that are flowing around it. And with the mycelium, I think is really applicable because as with Katya, so she's growing the shapes and these megastructures, and in the end, it's always something interesting. It's always something new, but still, you can see that this was grown by Katya. And I'm sorry, I forgot. There is a Rose who is playing with mushrooms as well, and a really big artist who's doing the Katya. You should
Which
know.
one? The one who does like chairs, or like the one who like there is like
I think his name is Ross, but
Philip Ross, his surname.
Philip Ross, yeah.
He was like one of the probably first.
Yeah, so in his works as well, look, you see, and you see that it's grown by him. So
yeah, weird aesthetics.
yeah, but anyways, so and you are just shaping an intention, and you can see you can predict the stages of the evolution of the project, and you can see how in every stage there would be something which get inspired you for the next project, or for the next stage to implement it in it.
Right.
Okay, so I think we have 20 minutes till the next lecture, so maybe we can have a break and then see you all again. And there would be a lecture from the point of urban design and the landscape urbanism, which is more macro scale. And then we will try to get acquainted with each other, which we started already, which is good. And a short introduction, and we'll try to get you inspired for the next session next day. So maybe we hope that you will start to think about the project and start to produce some drawings for the next step. Okay, so see you soon.
I just posted in the chat, just in case, the link for the next lecture, and see you soon.
See you all.
See you soon.
See you.
See you.